Welcome students once again. In continuation of chapter 6, last part of chapter 6 that we are going to discuss in this video. Here, in the previous video, I told you about germination, different kinds of different conditions for germination. And here in this video, we will see kinds of germination. As you can see here, I have noted down, I have written down here two kinds of germination. First one is hypogeic germination. The second one is epigeal germination. Alongside, I have drawn a diagram of a seed which is germinating. Now, this is the soil. When we sow a seed, we put the seeds inside the soil. And so this seed is inside, it was sown inside the seed, inside the soil. And here, as you can see, the seed is still inside the soil. The seed is not pushed above the soil, it remains inside the soil. It is because the structure of seed, I have told you, epicotyl and hypocotyl. You remember, epicotyl is the short stalk-like structure that connects plumule to the seed. And so, plumule is the part of the seed that grows into shoot system. So, the epicotyl elongates faster than hypocotyl here. And as a result, what happens? Epicotyl elongates faster than hypocotyl. So the seed remains in the soil. It is not in the case of epigeal germination. Here is hypocotyl. This hypocotyl elongates faster than epicotyl. So as a result, what happens as this hypocotyl is elongating, it pushes the seed outside the soil. That difference, I think, it is clear from the diagram. Here, the epicotyl is elongating before or faster than hypocotyl. So, the seeds are pushed downward, and so the seed remains inside the soil. Now, let's see the difference between hypogeic germination and epigeal germination. Number one, cotyledons remain underground. You can see here, cotyledon remains underground in hypogeic germination. On the other side, in epigeal germination, the cotyledons are pushed above the soil. Pushed above the soil. Second difference, epicotyl elongates faster than hypocotyl. Epicotyl elongates faster than hypocotyl. In epigeal germination, it's just the opposite. Hypocotyl elongates faster than epicotyl. Third, usually hypogeic germination occurs in monocot seeds, and epigeal germination occurs in dicot seeds. Students, do you understand? Two types of germination and the differences. Now there is another special kind of germination, known as viviparous germination. Viviparous germination. V-I-V-I-P-A-R-O-U-S. Viviparous germination. This is a kind of germination of seeds inside the fruit. The seeds remain inside the fruit and the seeds germinate inside the fruit itself. This is what is called as viviparous germination. An example in some mangrove plants. We find that seeds germinate inside the fruit itself. Now, last word that we are going to study is seedling. The ultimate aim of germination is the formation of seedling. You remember what is germination? Germination is the process of development of seedling from the embryo of a seed. Now after germination, the result is formation of seedling. What is seedling? Seedling is a stage of a tiny plant before it gets independent. It is a stage of the plant or a tree before it gets independent. That means before it produces leaves of its own and is able to produce food on its own. So before that, before it becomes independent, the stage of a plant after germination is called seedling. I hope you have understood this lesson and keep studying, write notes and send them back to me.